The human memory system functions as a dynamic, reconstructive engine, operating with far less fidelity than a perfect digital recording. This architecture means that vivid childhood scenes often populate our minds, even when the biological machinery required to record them was not yet fully functional. Historically, this gap in our record is called infantile amnesia, a phenomenon that has occupied researchers for over a century. Modern neuroimaging reveals that biology, rather than psychology, dictates the hard limits of what we can actually remember from our first years of life. Solving this paradox requires an investigation into the physical limits of the infant cortex, the biochemical vulnerability of how we retrieve information, and the evolutionary pressure to favor narrative over truth. The evidence suggests that the memories we consider foundational to our identity are often the exact ones our brain fabricated to secure our present reality. Infantile amnesia represents a critical developmental period where the brain is literally learning how to learn. This 2025 neuroimaging study from Yale used fMRI to monitor brain activity in awake infants, finally allowing us to see memory encoding as it happens. These researchers discovered that infants possess two entirely different memory systems. This diagram highlights the anterior hippocampus. In infants, this area is highly active, driving statistical learning, extracting patterns like a home's layout. Conversely, the posterior region, managing time-stamped episodic memory, remains inactive until 12 months. Patterns are stored, but specific events are not. These earliest years form an archival gap. The developing brain biologically prioritizes mapping the rules of the world over recording a personal diary. Postnatal neurogenesis provides a physical reset. This rapid wave of new neuron cell birth constantly sweeps through the brain. This aggressive cellular turnover remodels neural circuits, erasing the specific coordinates of early life events. A second constraint involves the delayed maturation of the prefrontal cortex. Lacking a mature prefrontal cortex, the infant brain cannot organize or archive long-term episodic data. Between the aggressive overwriting of neurogenesis and the absence of a prefrontal archivist, the brain biologically wipes its earliest episodic data clean. When an adult attempts to access an early childhood memory, the brain encounters this archival gap. However, the system's architecture is designed to avoid returning a null value. The hippocampus uses pattern completion. When the recurrent network receives a fragmented sensory cue, like the color of a toy, it forces it into an attractor state. It pulls from semantic schemas, generating a full image populated with plausible, constructed details. The adult hippocampus acts as a creative editor, rendering historical fiction from fragments to replace what was lost. Why do these fabricated scenes feel physically and emotionally identical to objective historical events? These cross-sections show the fusiform gyrus, which houses a neurobiological reality threshold. This region uses the vividness of a signal to judge whether an image is real. If the signal generated by pattern completion is intense enough, it successfully crosses this threshold. A mature prefrontal cortex would typically use source monitoring to tag this as an internal simulation. But because the gap exists before that cortex matured, the memory remains untagged. The brain trusts the intensity of the signal, permanently mistaking an internal simulation for external history. This vulnerability extends even into adulthood. The very act of retrieval physically alters the memory trace. This engram is the physical representation of a memory. Recalling it triggers memory reconsolidation, causing the synaptic connections to uncouple, becoming fluid and labile. External information, like seeing an old photograph, infiltrates the engram, wiring directly into the original trace before it hardens again. Sharing childhood stories in conversation triggers this reconsolidation loop, favoring shared consensus over objective truth. You are not remembering the original event. You are remembering the last time you recalled it, progressively hardwiring a social narrative into your biology. Why did evolution favor a memory architecture that so easily distorts historical data? The framework of active inference suggests the brain is a forward-facing predictive engine. It constantly predicts its environment, comparing predictions to sensory data. Mismatches generate error signals. To minimize these, the brain retroactively alters past memories, aligning them with the present to eliminate contradictions. Acknowledging a perfectly factual historical record is computationally expensive and offers little advantage if it leads to social isolation or identity fragmentation. Your sense of self, the I, acts as a fixed point, collapsing fragmented sensory data into a stable, survival-optimized narrative trajectory. Pattern completion and reconsolidation serve this goal. Our high confidence in a memory reflects its narrative utility, not its accuracy. Acknowledging the fabricated nature of memory is a recognition of the brain's mechanism for survival, the continuous, creative authoring of the